we're going to further derive this analysis to an important biological problem. That is the new gene evolution detected by genomic computation, which we find that actually new gene can serve as a driver for evolution of human brain. We know that today we human beings have a big brain. We also know that this big brain evolved not long time ago from our ancestor, which with smaller brain. During the evolution of brain size, our ancestor created a foraging tool indicate the origin of intelligence. Our ancestor also introduced fire, which is great event. And our ancestor also created a concept by painting, drawing in a rock, in the rock. Those show that our ancestor had ability to derive a concept with the growth of the brain. Most exciting today, we human beings know that where we are from, and also we know that what the universe looks like. So this observation immediately asks the question: what genetic changes occur in our ancestor drove evolution of the brain intelligence? Right now, let's analyze if the new gene play a role in brain evolution. Using a similar pipeline I just introduced in the last section, we identify the new gene in a group of vertebrate species using the principle as we showed in the insect, available everywhere like gene A, the old gene. But B only appear in 4 species, so we know that the B originated in this time, origin of gene B. And if a human being only have a gene E, then the single gene only appear of human being diverge from chimpanzee. All the rest of the gene C, D, F are very old. That by informatic analysis, we identify the new gene which originated in various evolution time period as I show in this red line, which evolved toward human lineage. On total, we identify more than 5,500 genes, which is mammal specific. Among them, there are about 1,800 genes, which is primate specific. So average rate of gene origination in vertebrate is about 20 genes per million year, which enter the genome of our ancestor. And in the primate species, then 30 genes originated per million year to enter the primate ancestor of human being. In our human-specific lineage, we have a highest origination rate, which is fixed about 80 genes per million years. Looking at the expression of this new gene using the recently created database of gene expression in different kinds of tissue in human being and mouse, we identify that there's a much higher proportion of new gene which express in the brain of human being compared to the mouse, while the mouse have different type of gene control their biology that you impact the distribution. Then we also compare expression level by compare expression of parental gene, and we see that after the new gene created, most often their expression level become up-regulation in the new cortex, and the prefrontal cortex we identify their proportion, and the proportion increased at the time evolution time approach to today. Very exciting discovery that we see that first, when we're looking at the distribution of those genes originated in different stages, as we show in red color, and all those genes appear in different stages actually expressed in the new cortex of human being. Surprisingly, they're not really expressing high abundance in adult brain, but they express in higher level in the prefrontal cortex and temporal lobe of the baby or early development of human brain. So this suggests that new genes evolved in the early development which will regulate expression of later gene and which will involve in the cognitive function. Here I give one example. A new gene in this 54 gene, which is human specific, called srgap2C, they evolved from srgap2 by partial duplication. Scientists express this gene in a mouse, and by compare to wild type mouse, which is in the mouse brain, those neurons actually look like this, which contain a certain structure here called the spine, which was involved in the connection between the different neurons. And the new gene srgap2C after expressed, and they observed that the spine have a longer neck, as we show up there, and also the more spine involved this gene in the brain of the mouse. So this suggests that with the expression of the new gene in mouse brain, it might help the neuron in the mouse brain have a tighter connection. Therefore, they might enhance the computing power of the brain, implicated importance of the function in the search of human brain. So in this section, we introduced that evolution of the brain was accompanied with the origin of new genes. Then we also found that new genes are upregulated in the new cortex, in particular the prefrontal cortex region throughout evolution of vertebrate. And finally, we report that many new genes, in particular human-specific new genes expressed in prefrontal cortex and temporal lobe of brain structure of the early development involved for cognitive function. Thank you.